This video is about the root nodule formation, what we call as nodulation. It's a process by which leguminous plants form symbiotic association with Rhizobium bacteria. We see in this association the bacteria induces nodule formation in leguminous plants, wherein this bacteria fixes nitrogen for plants and in turn gets supply of carbohydrates and other nutrients from plants. If we see the whole process of nodulation in flowchart, we start with a flavonoid release from root hairs, which mediates recognition. Second step is the root hair curling, which is induced when the nod factors are released by the Rhizobia upon binding. After that, we get the infection thread formation and its entry, which is followed by the bacterial entry. In the next step, we get the cortical cell response along with the nodule primordium formation. Then we have infection thread progression and Bacteroid differentiation, which is followed by nitrogen fixation and oxygen regulation. This oxygen regulation step is one of the important steps in this process. Then we have the nutrient exchange and finally the nodule maturation. Now let's see the whole process in detailed manner along with the animation. Here in this diagram we have the root hair. So when there is nitrogen limitation for the plant, first of all it releases flavonoids, which attracts the Rhizobium bacteria towards the root hair as shown in the animation. The Rhizobium has two important surface molecules for binding, the ricadsin molecule and glucomannan polysaccharide. On the other hand, we have the lectins on the root hair as shown in the diagram. Here in this diagram we can see how Rhizobium binds with root hair with these molecular interactions. So upon binding, the Rhizobia respond by producing nod factors. These nod factors are recognized by a specific class of receptor kinases on root hair that have LysM domains in their extracellular domains. The two LysM, that's lysin motif receptor kinases, are NFR1 and NFR5. Here in this animation, we can see the nod factors comes in and binds with these receptors. After that, we get the activation of signaling pathways and the root hair curling. In signaling pathways, we get the activation of cytokinin signaling and calcium spiking. In the meantime, infection thread starts forming as shown in the diagram. Now getting back to cytokinin pathway. The activation of cytokinin pathway induces cell division in the cortex shown in the diagram, which leads to formation of nodule primordium. The nodule primordium is the initial new structure that will eventually develop into mature nodule. Now in the next step, the infection thread advances and we get the Bacteroid differentiation, where we see the Rhizobia differentiate into Bacteroids. These Bacteroids develop capability to fix nitrogen. So in the next step, we see the Bacteroid has to fix the nitrogen now by the help of nitrogenase enzymes. But we have an excess of oxygen present there, which inactivates the Bacteroid nitrogenase enzyme. So to tackle this, the plant produces leg hemoglobin molecule. This leg hemoglobin molecule plays dual role. First role is the leg hemoglobin scavenges oxygen to protect nitrogenase enzyme in legume Rhizobium symbiosis. Secondly, by sequestering oxygen molecule, the leg hemoglobin creates a microaerobic environment within the nodule. This balance ensures that enough oxygen is available for the respiration of Rhizobia, providing energy for nitrogen fixation while keeping oxygen levels low enough to protect a nitrogenase enzyme. Then under these regulated conditions, we get the nutrient exchange between the plant and bacteria. Now at the end, we get the nodule maturation into different zones, with zone 1, zone 2, zone 3 and zone 4. These zones can be seen in this diagram. Zone 1, meristematic zone, where newly cells are formed. Zone 2, the infection zone, where Rhizobium are released and start differentiating. Zone 3, nitrogen fixing zone, where Bacteroids actively fix nitrogen. And Zone 4, the senescence zone, where old cells and bacteroids are degraded. And finally, through this symbiotic relationship, the plant provides a home and nutrients to the bacteria, while the bacteroids convert atmospheric nitrogen into a form that plant can use for growth. This incredible process ensures the plant gets the nitrogen it needs to thrive even in the nutrient-poor soils. So this is all about nodule formation and nitrogen fixation, a perfect example of nature's cooperation. I hope you like the video. If you like it, give it a thumbs up. Do consider supporting my work on Patreon or YouTube and make sure to subscribe to this channel. Thanks.